Okay, guys and gals. So just to see where we've been and where we're going, okay, in our outline, okay, the first thing I talked about are why do cells divide? So we can check that off as that has been done. Remember, there's two reasons they divide: to make more cells and because they just get plain old too big. Then we talked about karyotypes, chromosomes, which I didn't mention, but we did when we met Monday, that first week that right after Easter break. We talked about karyotypes and chromosomes and even chromosome mutations, such as duplication or deletion or a translocation or non-disjunction. And we can see those in the karyotypes. And we talked about karyotypes and all. And it was important to talk about karyotypes and to get to know really about those chromosomes, right? Then we reviewed mitosis and we paid special attention to the spindles and the kinetochores. That was during anaphase. Anaphase. So there were like new things that we talked about in there. And then in cytokinesis, we definitely talked about the two types of divisions that happen. This is like during telophase or cytokinesis, telophase slash cytokinesis, where we talked about that. Kinesis, yeah, I can't spell right now. Cytokinesis, thank you. Okay, then we talked about the types of asexual reproduction. Binary fission is usually seen in eucellular organisms. We also looked at the evolution connection. And I believe maybe I missed a page in there, but I did talk about budding, regeneration, propagation, but I didn't mention spores. Spores are just little cells that are, they can be haploid or diploid, actually, but they're cells that break off and eventually form. If they're haploid, you're going to have one spore fuse with another spore and then grow into the organism. And this happens a lot of times in fungi. Or the spores, if they come off diploid, they're just going to grow into the new organism right from that original spore. So that's where we've been and where are we going. So out of all of this that we talked about, I really want you to focus definitely on this and this, but make sure you know about those karyotypes and stuff, because you can't understand everything without doing that. And when we get into sexual reproduction, after we go through the cell cycle, that's going to be really important to understand, those terms: haploid, diploid, homologous chromosomes, alleles, some of those words that we mentioned there. So our next bit here that this is going to be on is the cell cycle. We're going to go over the phases and the checkpoints, and you might remember that from first year bio. But then we're really going to go into detail on the signals involved in moving that cell forward through that cell cycle. There can be chemical signals, physical signals, and genetic signals. Okay. And when those signals go wrong, that's cancer. Okay. And we'll talk about also when the signals all go right, and stem cell and differentiation, just very basic thing with that. So this is going to might take two videos, but let's hope not. Okay. All right. So here's our cell cycle. And think of it like kind of like a lifetime clock. If this was me, I'd be like, oh, little toddler Bonnie. Then little Bonnie goes to like elementary school, middle school, and high school. And then sometime during my midlife or young life, DNA synthesis happens. That's where you make like a copy of your DNA. I always say that's like me reproducing or something. And then old age is what I'm entering now. Okay. And then fortunately, because I made more of my own kind, they continue on. Cells are a little different. Okay. Cells, if we look at one cell and how it grows, this is that G1 phase, okay? It has a certain checkpoint. Kind of think of it as like a stoplight that says, are you sure you want to move on? And we're going to investigate these checkpoints a little bit more. If it does move on, it goes into the synthesis where DNA will be replicated. DNA synthesis, I wish they called it just DNA replication, because replication of the DNA happens here. And here's an interesting point. That G1 checkpoint, they often say, is the most important checkpoint because If a cell is not going to divide, then there's no need for it to move on to that S phase. Because think about it. Why would you need an extra copy of your DNA if you're not going to divide? And that's what our nerve cells do. Our nerve cells are perpetually stuck, and they call that G0. Kind of like if you're not going to divide, you kind of go off into this G0 phase where there's like no more division ever. Okay, and some cells are like that. Muscle cells are actually like that. Muscle cells don't divide a lot, and you would think, well, how do muscles get bigger? They just like grow, and they're microfilaments and stuff like that. Fat cells don't really divide a lot. You might be like, how do you get fatter then? The cells basically like fill up like water balloons, or they shrinky-dink down and stuff. And definitely nerve cells. If you damage any nerve cells, you know you're not going to be able to replace them. But skin cells, they're constantly going through because they're constantly being replaced, okay? So we have the S phase, and then we have more growth, and we have yet another checkpoint at the end of that G2 phase that's going to say, well, you know what? I know you did your DNA way back then, but I just want to check to make sure your DNA is all replicated okay, and that you have the proper number of mitochondria and Golgi and ribosomes, because we're going to have to divvy all that up. Then there's one more checkpoint that's in the middle of metaphase, and that's when that checkpoint looks to see that all the chromosomes are lined up. Now, I think this is said in the next couple pages. Let's hope so that I didn't have to write it down there. But yeah, there's a lot of writing here. Writing. Okay, but let's take a look at it. It's the sequential events in the cell cycle. Yeah. There's both chemical controls, we're going to talk about them, the cyclins, and physical controls, okay, actual like touch and stuff like that, physical things. Checkpoints, we mentioned the checkpoints, okay, mentioned the G1 checkpoint seems to be the most important one, okay. If it receives the go-ahead signal at G1, then it will eventually complete the S, the G2, and M, because if a cell, and let me like go back and look at the diagram with that. So again, if that cell decides to go past that checkpoint, it will go all the way on until it gets through S, G2, and M, or it will die. Apoptosis, remember that, okay? So G1 is very important, but it will complete those other ones. But it will also look, and here's where that GO is mentioned. If it doesn't go on, it goes into that like offshoot called G0, where it's not going to divide, and that's like your nerve cells. Let's mention that. Nerves, fat, muscles. It's so weird to think that they don't divide much, but skin cells are definitely not in the G0. Okay, see what it says on it. Aha! Now we got to get into those signals. These are the chemical signals. Okay, remember we said to get through that cell cycle, there's going to be chemical signals, there's going to be physical signals, and there's going to be genetic signals. Okay, so what are the chemical signals first? And these are probably the most important. Yes, they are controlled by genetics, and we'll get to that in a little bit more detail, because obviously genes need to make these things. And it's really all about these things called cyclins. Cyclins, and there's those kinases again. Do you guys remember about kinases? They're the ones that are enzymes that break the phosphate off of ATP and cause an action, okay? So cyclins are going to work with kinases to get things to move along. Remember, kinases are always like causing something to move along, all right? And how does it work, okay? Well, these cyclins are going to make these CDK complexes. What does that mean? CDK stands for cyclin-dependent kinases. Cyclins and kinases will combine to make this complex. And when they combine, they can move the cell past those points of in the checkpoints. Okay, so let's take a look at first here. Okay, and we'll start with G1. All right, during G1, we know that the cell is growing. Okay, so we have a lot of growth happening here. Growth. of the cell, and the cell's being its regular cell self. It might be eating and digesting, pooping, and doing all the stuff that cells do. But it's also starting to build up its CDKs, its cyclin-dependent kinases. In other words, cyclin is being made and kinases are being made. Okay? All right? And so we have this complex that's starting to build up. Okay? Now, if we look at this up here, okay, let me just stick with this first, okay? These cyclin-dependent kinases build, okay? And there's different factors that come in that kind of move them through certain things. What this one is showing here is the one that's going to move them past the G2 checkpoint. Yes, there's a type of MPF happens to be the type of cyclin that's going, or the type of protein that's going to hook up with the cyclin-dependent kinase that gets them through G2. That will increase until eventually a certain other cyclin called MPF kind of like gets activated. When these two come together, then it can move past that. But there's another one that's also happening that will move it past the G1. There's one that moves it past the G2, and there's one that moves it past the M phase too, that other checkpoint. Remember we said that there's a G1 checkpoint and there's an M checkpoint. This graph and this here only are pertaining to the G2 checkpoint. And MPF, right, which stands for maturation protein factor, I think it's called, is only like one that's been greatly studied to get it past the G2 phase. Okay. And what's interesting is this CDK, this complex, this CDK, which is the cyclins and kinases, they kind of always maintain a certain level in the cell. Okay. But they will increase in their levels when this other cyclin, MPF, seems to come around. Okay, so we see like how this like fluctuates. When the other cyclin comes around, then we see more activity happening. Okay, but the CDKs are pretty much there all the time, just getting ready to capture this. Okay, so that's where these cyclins and these CDKs, which are other cyclins and kinases, all kind of get involved to move past those checkpoints. Okay, so yeah. And there's, besides these cyclins, these kinases, and these other cyclins, there's also all these other things called growth factors. Growth factors. There's some 50 different growth factors that are also. So when it comes to chemical signals, those chemical signals are going to help them get past those checkpoints. And MPF is just one such chemical signal that gets them past the G2 checkpoint. But there's another one for G1. I can't think of it off the top of my head, and another one for the M phase as well. But those cyclins will attach to the cyclin-dependent kinase, which is more cyclins and kinase combined. So you have cyclin upon cyclin and kinases. When they both attach, that's when it gets through that point. But there's another one that gets them past G1 and another one that gets them past M. And those are not growth factors. Those are cyclins that do that. Growth factors are something else, chemical that can be released by the cell to also promote that cell to get through its cycle. And why do you want the cell to get through the cycle? So it'll divide. Okay. Now remember, you don't want cells to divide if they're like messed up, right? Because then that's cancer. Okay, so we'll see where that comes into play later with cancer. So those are some chemical signals, growth factors and cyclins. Cyclins that attach to more cyclins and these kinases that get them through the checkpoints. Okay. As for the physical things, these are a little easier. There's the density-dependent inhibition, which basically means what it says. It's dependent on the density. If the density is high, it inhibits it. Okay. So think, like kind of like if we have too many kids in the class, then we got to kick some out down to the cafeteria, okay? Well, that's the same with cells. If you have very few cells in an area, they will divide. But once they fill up an area, they're not going to divide anymore. Take some out and they'll divide. Same like a cut. Take some out and they'll divide more. The other one is anchorage. Cells need to be anchored to something solid in order to divide. So not only do they need their space, but they also need to be attached to something. And those are the two physical signals. Okay, now this video is going to end, but I'm going to talk about the genetic signals after that. Okay.